Assalamualaikum. DNA extraction is the process of obtaining pure DNA from a sample through three basic steps, which are cell lysis, protein removal, and DNA purification. We will do demonstration of DNA extraction. I am FAZIHA and I will start it first. The microcentrifuge machine is used to separate the mixture of cells and medium. In this experiment, we use the samples of bacteria cells, which is E. coli. The collection tube, pipette tips, and the micropipettes. We can adjust the volume that we want to pipette in. There are two stops. Press leisurely for the first stop and forcefully to the second stop. Usually, the color on the top is compatible with the same color of the pipette tips. Firstly, insert the pipette tips. Pipette in 1.5 ml of E. coli to extract its DNA using the pipetting technique. Pipetting aseptically under the laminar flow in order to prevent the bacteria culture from contamination. Next, pipette out into the tube. Bacteria culture need to centrifuge in microcentrifuge machine because the cell is mixed with the medium. So, we want to separate them by spin down the cells to get the only cells. The bacteria culture is put into the microcentrifuge machine in opposite position and spin for 1 minute in 12,000 RPM. At high speeds, a centrifuge can easily become unbalanced if equal masses are not located opposite each other, and it will cause the damage to microcentrifuge machine. So, this is bacterial culture after centrifugation. There is two phases. Phase 1 is supernatant, which is the remaining solution that lies above a precipitate, while the bottom part is pellet, which is the small and compressed sediment.
NURSYAZWANI from Group 2 and I will proceed for the next step. In these steps, we will use proteinase K and lysis buffer solution, microtube, water bath for incubation, vortex machine, and micropipette. After discarding supernatant completely, by using micropipette, take about 100 microliter of lysis buffer and transfer it into the microtube that contain the cell pellet. The purpose of lysis buffer addition is to break down the cell wall of E. coli, which is the bacteria that we use in this experiment. After adding lysis buffer, the microtube was put onto the vortex machine to ensure the cell pellet and the lysis buffer were mixed up well. After vortexing, pipette 20 microliter of proteinase K into the microtube to degrade the proteins and incubate it in the water bath for 15 minutes at 55 degrees Celsius. For every 5 minutes of the incubation, the microtube should be flipped to ensure a proper mixing of the solution. However, if the solution is still not clear, the time for incubation should be added for another 30 minutes for a proper mixing. After 30 minutes, the solution is ready for the next steps.
After incubating at 50 degrees Celsius for 15 minutes, the next step is to add RNase-A. So why do we need to add RNase-A? This is because RNase-A is used to degrade the RNA. Since this is DNA extraction, we only want the DNA and not RNA. This is RNase-A. We need to pipette out 20 microliters of RNase-A and then put it inside the bacteria. Vortex the solution to mix it in a vortex machine. And then we can leave it at room temperature for 2 minutes. Okay, so we have everything added here: the lysis buffer, the cells, the RNase-A, the proteinase K, and binding buffer. So we need to transfer this solution into this spin column by pouring it. Make sure every single drop is poured into the spin column. And then we have to centrifuge this for 30 seconds at 12,000 G. Okay, so assume it as a spin column. This is spin column. It comes in two parts. The upper part is spin column and the lower part is the collection tube. Usually, on the spin column, there will be white area on the bottom of it. That is where the DNA will be trapped. By combining these two and centrifuge it in a centrifugation machine, the DNA will be flowed into the collection tube during the centrifugation process.
Assalamualaikum. I am NURUL SYAFIKAH binti Darul Aludin. I will continue the procedure of binding buffer centrifugation step. After all the solution is transferred inside the spin column, combine the spin column and the collection tube together and centrifuge the whole thing in the microcentrifuge machine. The machine will spin at 12,000 RPM for 30 seconds. As a result, the unwanted debris, cell wall and buffer will flow through into the collection tube. Then, take out the collection tube and degrade the solution. Now, put back the collection tube with the spin column together. Next are the cleaning and washing buffer step. This involves the use of two solutions which are clean buffer and wash buffer. By using the micropipette in the cleaning buffer inside the micropipette and then put them into the matrix column. Then centrifuge them in the microcentrifuge machine at 12,000 RPM for 30 seconds and discard the flow through. The same procedure are using for washing buffer step, but do it twice, which is put in 500 microliter of wash buffer into the matrix column and then spin them in the microcentrifuge machine at 12,000 RPM for 30 seconds. Discard the flow through. Next is the crucial step, which is spinning the empty column step. Centrifuge the empty column at maximum speed in the microcentrifuge machine for 1 minute. This step is crucial because there will be some excessive ethanol inside the column. So we want to get rid of that excessive ethanol because the ethanol will damage the DNA in a long period of time. Lastly, the final step, which is elution step. 50 microliter of elution buffer that preheated to 65 degrees Celsius is added into the matrix column by using the micropipette. Then, incubate them at room temperature for 2 minutes. Then, centrifuge at 12,000 RPM for 1 minute to elute the isolated genomic DNA. So, here is the DNA that was eluted into the collection tube. Throw away the spin column because the DNA is already in the collection tube. This is the pure DNA that we want. Ensure to keep the DNA at negative 20 degrees Celsius. That's all from us. Thank you.
That's all from us. Thank you.